So there are over 4,300 known species of aphid that feed on a variety of different host plants and have different diet preferences. So of that 4,300 species, only about 300 are agricultural pests, meaning they cause damage to different crops. But of those 300 species, they're spread all around the globe, so they damage a lot of different economies and different agricultural areas. So aphids reproduce mostly asexually, which allows them to have high fecundity and a very short generation time. So they go through what is called telescoping of generations, which means that when the first aphid clone is born, that clone is already pregnant with the next aphid, which is really fascinating to me and just allows them to just explode quite quickly. So I used 5 different aphid species for my project, 3 of which were specialists. So specialist aphids feed on one specific host plant family and potentially only 1 or 2 species within that family. So I used Aphid, which is the bird cherry oat aphid. I'll be referring to each aphid with a short letter acronym just for easiness. So bird cherry oat aphid is BCA. I also used Apissum, which is the pea aphid, and U. nicrotuberculatum, which is the red goldenrod aphid. So Aphid feeds on maize, which is a monocot. The pea aphid feeds on fava bean or other legumes, which is a dicot, and then goldenrod aphid feeds on goldenrod. So just like there are specialists, there are also generalist species of aphids. Generalist species can feed on lots of different host plant families and many species within those families. So the specific generalist species I used was Mysis persicae, which is the green peach aphid. She comes in 2 different subspecies, which is green peach aphid pink, which is tobacco adapted, and green peach aphid green, which is non-tobacco adapted. This specific generalist species can feed on over 40 different host plant families, and she is spread all across the globe, which makes her one of the most damaging generalist species because she is able to feed on so many different things. So even though they can feed on a variety of different host plants, that doesn't mean that they don't have preferences. So one thing we looked at was host plant acceptance. So we used 6 different model host plants to feed GPAP. We used maize, which was our monocot representative, Erysimum, fava bean, Arabidopsis, tobacco, and N-benthmiana, but specifically the ASAT2-1 mutant, which removes the acyl sugars off of the plant. We had to use a mutant species because the acyl sugars make it really hard for the aphids to be able to colonize on the N-benth. So in order to get a proper infestation, we had to use the mutant species. So aphids feed by weaving their specialized mouthpart, which is called a stylet, between the different plant cells and tap into the phloem, which is a nutrient transportation system throughout the plant. They go through sucking and spitting. So while they're sucking in that special phloem nutrients, they're also spitting out unique compounds back into the plant, which are known to dampen the host plant defense systems. So a lot of compounds that have been studied in the saliva are the proteins. They are known to impact calcium ion signaling and also help the phloem callus when they detect invasion. So spitting the protein into the plant help it to not callus so that aphid is able to continue feeding. One thing that we wanted to look at are do metabolites in the saliva also impact the plant in the same way? We know that protein composition in the saliva changes with different host plants and across different species. So we wanted to test that hypothesis, but with metabolites. So for my hypotheses, I believe that GPAP, the pink tobacco adapted subspecies, would have the most aphid fitness or survive the most and have the largest adults in Nicotiana tobacco, which was the tobacco plant, just based on previous observations by my mentor, Hilary Fisher. I also hypothesized that the different aphid species would share a set of core metabolites, but they would vary in concentrations and type across the different species and diets that I fed them. So in order to look at my first hypothesis, we conducted a bioassay. So using the 6 different host plants that I mentioned earlier, we placed about 4 or 5 adult GPAP aphids in each of the different cages on the plants. We let those aphids proliferate and have roughly around 10 juveniles. At that time, when they had about 10 juveniles, we took out the adults and thinned down the juveniles, and then let those juveniles grow into adulthood over a course of about 9 days. We then took off the now adults and froze them for further measurements and counted to see how many aphids had survived. So for the measurements, we actually used 1% agar to plate the frozen aphids on, so they would be in a nice area to take pictures. And you can really see a difference even with the naked eye. So although these were all the same exact species and they're all adults, they're vastly different in size just based on what they were feeding on. So we were able to image each individual aphid with a stereo microscope and take really high quality pictures with a scale bar. And then I was able to feed those into image J and highlight just the individual aphid and find its area, and then make some really cool comparisons with that. So based off of our bioassay, we determined that N-benthmiana, which is this one with A right here, had the most aphids survive on it, which was actually a statistically significant result as compared to the rest. And they also had the largest aphids, which meant that they were getting the most nutrient out of the plant because they were able to feed the best. So because of that, I would move to say there was not support for my first hypothesis that they would perform the best on Nicotiana tabacum, but instead they performed the best on the Nicotiana benthamiana ASET2-1 mutant. So to test my next hypothesis, we had to collect saliva from the aphids. So to do this, we first reared the green peach tobacco adapted subspecies on the 6 different host plants. We let them grow for about 3 generations so that the aphids that we collected had only been on that host plant. They hadn't been previously fed on something else. We put those in different saliva cups, and we also did the 4 remaining species. We made cups out of those. So how you make a saliva collection cup is you put the aphids, roughly the same amount for each species or each host plant, in the cup, and you sandwich 100 microliters of HPLC grade water between 2 layers of parafilm. That creates kind of like a pseudo phloem for the aphid to be able to stick their stylet up through and suck out the water and hopefully spit back into it. So then after about 24 hours, we collected that water and ran it through liquid chromatography mass spectrometry to generate chromatograms. So you can really see a difference here. This top is aphid saliva, and those really cool peaks are actually unique mass features that were found in the aphid saliva. But when compared to the negative control of just water, you can see that there's a large difference in the peaks present. So we actually took anything that was represented in the negative controls and filtered it out of our chromatogram samples, just for anything that could have been from the parafilm. But then once everything was control filtered, we compared all the chromatograms and what peaks they had in order to make some comparisons between how the saliva composition changes with diet and species. So we had a very large data set. So just to pull out some really important things and just to orient you, this is still all samples from GPAP fed on the different host plants. So we noticed that there was about a 10% overlap between all the different host plants, which was really interesting. So they all shared a core percent of metabolomic features. Tobacco had the highest unique percent. So those were metabolites only found in saliva from GPAP fed on tobacco. They weren't found in any other host plants. Arabidopsis was also relatively high. Erysimum was very, very low, which was pretty interesting. And then benthamiana and tobacco, although they share the same genus, there was not a large overlap. So that's something that might want to be looked into for future discussion, because we're not quite sure why that overlap was so low. So we pulled out 2 really important metabolites from that 10% overlap. Citric acid and malic acid are both known to be calcium chelators. So this can help disrupt that calcium ion signaling channel. So this is why we wanted to look at them in order to see if those metabolites were present and if that could be what their purpose was. So from this, we can see that tobacco had the highest amount of citric acid as compared to the other host plants. But for malic acid, we didn't have any statistically significant results. So this can show that host plant does impact the composition of the saliva, but not as much as we may have thought originally. So we made a lot of the same comparisons for the different host plants. So again, just to orient you, GPAP, we decided to use the tobacco-fed GPAP just as our representative because it had the most unique mass features. So we can see here that, again, almost 10% was shared between all the species for metabolomic features, which is really interesting, because this could lead to discussion that aphids share a set of core metabolites no matter what they're feeding on or what lineage they come from. GPAP had one of the highest, and I believe this could be because she's a generalist species. So she needs to learn how to infest a lot of different plants, so she has to have a lot of unique things in her saliva. The GPAG, the non-tobacco subspecies, was a lot lower in percentage. We also noticed that there wasn't quite a very large overlap between the 2 species. This is also something that might want to be looked into further in the data set, because we're not sure why this has occurred as well. And P. aphid and GR aphid, which are both specialist species, they also had quite a low overlap. This may be because they're trying to infest completely different host plants. So we pulled the same 2 special metabolites out of that roughly 10% overlap in the middle. And this one, we could see a lot more statistically significant results. So for GPAP, it had the highest abundance of citric acid in its saliva as compared to the other species. And then for malic acid, it was actually the P. aphid followed by the goldenrod aphid that had the highest amount of malic acid per percentage in their saliva. This could show that although host plant did not have quite as much of an effect on the amount of malic acid in the saliva, species does play a larger role in that, just based on what lineage they come from or what they're trying to feed on. So because of this, I would move to say that there was more support for my second hypothesis, that although these aphids are feeding on completely different plants and coming from different lineages, they do share a set of core metabolites that probably just aids them in infiltrating any plant. So in summary, we saw that host plant does play a major role in adult survival and aphid size as impacted by the bioassay. We also saw that both host plant and species impact the metabolites present in the saliva, although species may play a larger role. And aphids do seem to share core metabolites, but the relative quantities and the order in which they are highly abundant does often vary. So for future research, when we ran through our chromatograms and found the most abundant peaks, there was a lot that were highly abundant but unknown mass features. So we're not quite sure what that compound is that's running out. But it would be really interesting to look and try to discover and identify what those unknown compounds were. We can use something that's a GNPS interface. It's kind of like a spectral library that matches up the MS2 spectra to other ones that people have run through before. And it helps give you an idea of what your compound may be. We also could compare saliva from a different generalist species, even the green peach aphid green subspecies, feeding on the different host plants to make further comparisons, and also just further teasing apart our very large data set and making more comparisons, just like the ones I mentioned earlier, like trying to figure out why things that we thought should have a higher overlap had a very low overlap. And with that, I would like to thank everyone that's helped me personally and professionally this summer. I could not have done any of this awesome research without you. And I would like to thank you all for listening. And at this time, I'll take questions.
Thank you for your presentation. I enjoyed it a lot. You work with aphids.
I do.
How do you keep them contained? I know that aphid infestations were a problem for my lab throughout the summer, but how do you keep your aphids contained? And then do you think the way you do that affects your results? Like if you could work in a more isolated facility, you would be able to get more accurate results or other things? Like how does the containment affect the experiment?
Yeah, great question. So we try to keep our aphids from cross-contaminating a lot because obviously that could really interfere with your results. So actually down in the basement is our aphid cave. We have, if you've ever seen those like bug catchers that they're kind of like nets that have zippers to open. So we keep the host plant inside that and put the aphids in that. The aphids aren't able to get out unless you left the door open. So we try to keep each different replicate and everything isolated, especially with the bioassay. We had cage clips that you put the aphids in there that they're not supposed to get out, that we did have some tragedies where they did escape. And so we threw out those replicates, but we try to keep them isolated, at least down in the basement. And then for our saliva cups, they're covered in parafilm. And although they're stabbing it with their stylet, they're not making holes that are large enough for them to crawl out. So you're very accurate in saying that that could mess up results, but we try to be very careful. So
thank you.
Lydia, great talk.
Thank you.
So you might have already actually gotten this question before, but why did you use the one plant that you had to mutate to stop producing the acetyl sugars if it's not found in the wild? So why did you choose that plant in particular?
Great question. So why did we use Enbenthmiana and why did we use Enbenthmiana mutant? There are two different questions. So we use Enbenthmiana because it's a really good model plant and it's good for gene editing. And so if we want to do further experimentation to see how aphids are really interacting with that plant, it would be good to choose that. But we had to use the mutant type because to get that proper infestation, we had to remove the acyl sugars because that's a plant defense mechanism that the aphids aren't necessarily able to overcome as well if we wanted to get proper results and have good comparisons between the different plants.
Thank you.
Thanks, Lydia. That was amazing. One thing I noticed in your data is that the tobacco plant had like the most diverse group of chemicals that it wasn't seen in other plants. Do you believe this may have to do with tobacco's native plant defense mechanisms, kind of like you remove the acyl sugars on the other plant? Do you think tobacco has some sort of special defense mechanism against aphids that aphids are trying to overcome that we're not aware of yet?
Definitely. So tobacco obviously has nicotine in it, and the GPAP, tobacco adapted subspecies, is able to combat that plant defense mechanism. So we think that that's also why GPAP had a higher percentage of unique metabolites because they are working to overcome that. So GPAG, the non-tobacco, they're not able to overcome that nicotine. So that's why they have less metabolic unique features. That makes sense.
Thank you.
Thank you.